Hello, I am grateful to all of you who are attending and taking time out from the schedule. And we discuss about clinical immunology. Basically, which function immunology system. First of all, kill the pathogen, and secondly, don't harm the host. That is everything. But sometime something wrong and couldn't kill the pathogen and resultant go infected and sometime harm the host and develop disease, first of all autoimmune disease. I like framing lectures with questions and then use the lecture to answer, and at the end we'll summarize and make sure what we answered. And so, first question: why do you get some infected, like chickenpox only one? How can we generate immune system that could recognize a broad array of pathogens and with incredibly high sensitivity and specificity use immune system of coding DNA? Why do subsequent immune response to a pathogen occur more rapidly and at higher titers than previous? How does the immune system provide a high degree of sensitive and specific to broad array of pathogens without attacking self? Why are T and B cells effective against different pathogens? Pictured of this David Wetter Boeing bubble that some of you may be familiar with David Boeing bubble. What is amazing about David? He was born without functioning immune system in terms of B and T cell, and his two older siblings died within the first year of life from infection. Infections were unusual. Pretty much figured out that they had an immune deficiency. When David's mother was pregnant with him, scientists said all likelihood is that he may have the same disease. If we let him come out the same way, this rather the chance where he would die in the first year. Was brilliant idea. Well, if you don't have functioning immune system, you can't protect yourself from pathogen. But will protect from pathogen was born through caesarean section and basically lived his entire life in the plastic bulb. All his air was filtered. Most food was autoclaved. He grew up in his bubble, but when he was about 16, the scientists presented him with an option of getting a therapy of bone marrow transplant from donor to reconstruction immune system. Unfortunately, he died too as his brother sibling. The most traumatic infection that mankind has ever seen is smallpox. They killed the billion people and is incredibly dramatic. This picture displayed and dramatic clinical picture. 30% people was died, 70% of survived, and was noticed that people that there was smallpox only got it once. Therefore, this was called immunity. The advance in smallpox was Edward Jenner, famous doctor from England. He was smart. He made observation. He observed that women who were functioning as milkmaids would get this infected called cowpox. That was similar to smallpox, but instead of getting hundreds of lesions, they only got a handful of lesions. But these individuals never got smallpox. Edward Jenner has questioned why don't they get smallpox. He took crusted lesions from the cowpox individual, injected actually a young boy with it, and turned out the boy was protected from disease, and smallpox vaccine was generated. This picture describes eradicated of smallpox in the world by vaccination, officially irradiated up to 1980 year. Edward Jenner didn't know about B cells, T cells, antigen, and another immune response. He just had a brain and used his brain. Designing the immune system. Present as the innate immune system, acquired immune system, and barrier organ. Barrier organ is skin, mucous immunity, soluble protein as an innate immune system present, complement, a lot of protein production, liver, acquired immune system production, antibody, cells as an innate immune system present, phagocytosis, macrophages, dendritic cells, Langerhans cells, and acquired immune system have a T and B cells. Mediators, interleukin, a lot of number, tumor necrosis factor, subtypes alpha, beta, gamma, and gamma interferon as acquired immune system. Which functions have immune system? Three higher are. First are recognized, foreign and self. Accentuated foreign, of course. Secondary are reacted. Recruitment effector cells and eliminated pathogen. And remembered. This is important for a secondary attach with antigen. Target of the immune system, extracellular bacteria, parasites, and fungi. For instance, streptococcal pneumonia, Clostridium tetani, Trichopanosoma brucell, pneumocide carinii, respectively cause pneumonia, tetanus, sleeping sickness, and also pneumonia. Intracellular infected mycobacteria, it gets inside macrophages. Antibody can't do anything. Antibody be able to get virus be useful before they get inside. And finally, parasites, warmth. T cells are small molecules performed into parasites, but parasites laughing like, you know, give me a break. Is that the best you can do? Another portion of knowledge, what is immunogen? Anything that the immune system generate and immune response. And antigen. Antigen and immunogen is not equal point because sometimes substances have antigen function but not able to take immune response. This present innate immune and adaptive immune system present different cells. For example, innate immunity present macrophages, neutral killer, neutrophils, eosinophil, basophils as a blood stream, dendritic cells and tissue present, and separate between innate immune system and adaptive immune system cooperated T cells and natural killer. B cells present only acquired an adaptive immunity as activated plasma cells and production antibody, and T cell split to different cells CD4 and CD8 have different function, of course, and inactivated action each other. This picture about our question for why why do subsequent immune response to a pathogen occur more rapidly or and at higher titer than previous type? Primary antigen expressed from first day up to 7-10 days appear production immunoglobulin M as a first immunoglobulin. Through a few days, immunoglobulin M switched to production immunoglobulin G, and concentration immunoglobulin decrease rapidly to normal. Up to 3-week disease, we have a lot of concentration immunoglobulin G, enough to defense from this infection. If people have secondary antigen exposed, similar antigen, this immune response have more speed and immunoglobulin M appear more earlier than 7 days and switch it to production immunoglobulin G earlier too, and have a more higher concentration type immunoglobulin G. Next picture reflects difference between T and B cells, difference between them receptors. B cells have important activated receptor CD14-18 and another different time. T cells have some similar receptor. About this one, remember cells, this present receptor CD2, CD3 from the cells beginning from thymus production, T cells receptor and adhesion molecules different only between CD4 as a helper cells and CD8 as a T cells cytotoxicity. This picture difference and get our understanding difference between B and T cells cooperation. They have some similar structure as region. For example, antibody isotypes have variable region, variability region, and T cells have antigen binding and variability region too. And constant region have as antibody, as constant region have T cells too. But different between B cells ask, I attack invades outside the cells. T cells, I attack invades inside the cells. And antibody have different subtypes, for example, M, A, G, E, and have different size and structures. Monomer have two FAB fragment and FC fragment. Immunoglobulin E involve only allergic reaction and functioned immediately type allergic response, dimer and pentamer. And T cells present as T alpha beta T cells have more high concentration into the blood, up to 85%, and CD4 gamma delta up to 50% between all of them. We continue discussed later.